Good afternoon one and all. Myself Arjun Tirdal. Today I am going to present on plant microbe interactions in soil, enhancing soil health and crop production. The abstract of this project is the study explores how soil microbes like Rhizobium arbuscular mycorrhizal fungi and Pseudomonas fluorescens improves agriculture productivity in soil health, just like enhanced plant growth, improved soil health, increased stress resistance and reduced reliance on systematic fertilizers. Introduction of this is these microbes form complex relationship with plants which are crucial for soil and health crop production. Benefits of plant microbe interaction: it helps in nutrient availability, promoting plant growth and disease suppression, improving soil structure. The objective of this project is to investigate this symbiotic relations between soil microbes and plant roots in agriculture ecosystem, to assess the influence of beneficial microbes on soil structure, fertility and nutrient cycling, to examine the potential of plant microbe interaction in promoting sustainable and organic farming practices. The methods and the material involved in this project is rice, wheat and maize, and the other data collection from other sources like CGIAR, USDA. Limitation is it is a previously published in microbial behavior can vary with the soil type, climate and crop species. These are the results: symbiotic relations between soil microbes and plant roots. The Rhizobium using nitrogen fixation, it helps in increased nodulation availability of in plant roots, and PGP plant growth promoting Rhizobium, Sidophore production hormone modulation mechanism. It helps in better root colonization and stress tolerance. And other influence of beneficial microbes on soil structure: zinc solubilizing bacteria, it solubilizes zinc, binds and salt particles in soil, improves 22% of water retention plus 38% of enzyme activity, and microbial consortia, nitrogen fixation, respiration, myelinization. It helps in fertility index raised from 7.4 to 9.3. Based on this, we can discuss about the virtual project on plant microbe interaction, highlights their crucial role in agriculture ecosystem. The study reveals how these interaction, particularly symbiotic relationships with nitrogen bacteria and mycorrhizal fungi, are fundamental in improving soil health, nutrient cycling, crop productivity by enhancing nutrient absorption, improving soil structure and boosting plant resilience to various stressors. Beneficial microbes offer a sustainable alternative to synthetic inputs. And we can conclude that this study explores how harnessing soil microbiota like nitrogen fixing bacteria and mycorrhizal fungi can lead to ecological productive gains. The project concludes that plant microbes interactions are the foundation for sustainable and soil fertility, reducing dependency on chemical fertilizer. It highlights on that microbial community enhances soil structure, boost crop resilience, paving the way for paradigm shift towards regenerative farming. Thank you.
My topic is about isolation and biochemical identification of the bacteria from the bovine mastitis cases. Introduction: the milk which is obtained from the animal which is suffering from the mastitis is called as mastitis. Mastitis milk. The mastitis is the inflammation of the mammary gland which is occurs in the dairy cattle. It is also infect the udder tissues, in which the milk udder of the dairy cattle is become hard, firm and the mastitis milk is generally having a lower solid non-fat casein and the lactoses. Milk contain the higher acidity rate and the higher pH. the mastitis also affect the public health, in which it contains the environmental bacteria like E. coli, and it also contains the Staphylococcus aureus and Streptococcus agalacea bacteria. The etiology, the bacteria that has been causes the mastitis in dairy cattle, which includes the E. coli, Staphylococcus aureus and Streptococcus agalacea. The signs and symptoms of the mastitis diseases, which includes the affected udder may be the swollen and hard to touch, and sometimes it is firm and warm. The milk which is produced by the infected animal, which contains the higher pH and higher acidity rate and having off odors. It also having the symptoms like reductions in the milk production and increase in the body temperature of the dairy cattle. The study design and the materials, I have collected the 45 milk samples from the veterinary hospitals in Gadag, and I have streaked these milk samples in the differential media, which includes the blood agar, in which it is used to grow the gram positive bacteria like Streptococcus agalacea, and also the McConkey agar, which is used for the growth of gram negative bacteria like Enterobacteria species, which include E. coli and Klebsiella. And mannitol salt agar, it is used to grow the gram positive bacteria like Staphylococcus aureus, and it is also, this media is also used to differentiate between the Streptococcus and the Staphylococcus species. And eosine methylene blue agar plate, which is helpful for the growth of the gram negative bacteria like E. coli, in which the E. coli grows in this agar plate media, which gives the blue color metallic shield on the plate of the agar. Diagnosis of the milk sample, which is include the rapid platform test, that is California mastitis test. This test includes the CMT reagent and the pedal plate. Bacterial isolation, the milk is inoculated in the differential agar plate, like on the mannitol salt agar, the Staphylococcus species grows and gives the yellow color occurrence on the Staphylococcus aureus, and the on McConkey E. coli grows like pink colored colonies. And upon identification and isolation using the gram staining method, these isolates are undergo gram staining procedure, in which when viewed through the under the microscope, the gram positive bacteria are obtained like purple color cocci, and the gram negative bacteria are occur pink like bacilli. And biochemical tests are performed to confirm the Staphylococcus aureus and the E. coli bacteria, which include IMVIC test, indole, methyl red, Voges Prosker test, citrate utilization test, and oxidase and catalase. Catalase is used to confirm the Staphylococcus aureus species, and IMVIC test used to confirm the E. coli bacteria. Indole test, which gives you the red color ring, which indicates the positive for E. coli species, and MRVP gives the red color positive, and Voges Prosker VP test, which gives the crimson red color, which indicates positive for E. coli, and triple sugar iron, in which the E. coli species are obtained like yellow butt and yellow slants, and citrate gives the negative result for the E. coli. These 5 tests confirm there is the presence of E. coli species, and last one catalyst test performed to confer the Staphylococcus species, in which H2O2 is used, 1 drop of, and culture is added, which gives the effervescence like bubble formation, which confirms the presence of Staphylococcus bacteria.
Yes, thank you. I would like to congratulate everybody for your grand success, what you have made today, and submitted your presentation, presented your work in front of our judges. Twice he had called to ensure that everything is going smooth. Even though he is absent, but he is always present here with us, with his vision. Professor N. Raju sir, for gracing this occasion and making this event. On behalf of the department, I request Manju to present vote of thanks and token of appreciation. Now I request Raju sir to say a few words about our department and about our students.
I am jealous also. We are happy then. So, should I speak? This is microbiology related seminar. I am not a microbiologist. Not expect very high quality research since you are working or maybe for various constraints. We cannot expect. But really I appreciate the initiation and whatever you have taken. I really. They are friends or what? Both. Then? Brain, then?
Now I request Devendra madam to give her input.
Lacking and all that, where I can collaborate, where I can work. So I don't have a hand like Syed who can join hands with me and flat up things. So I was just wondering where all you are a light batch, but I can definitely say you are all star players now. We are winding up the subjects, you know. In another 1 year, we will be winding up the subject. Even I am one of the stakeholders. Even sir is also one of the stakeholders. So these beautiful subjects which were offered online or distance mode, this one, you take degree or not, that is a different story. Degree is not at all the issue. Second thing, as sir was telling, microbes, gut microbe, it is one of the serious and important in your future endeavors. Thank you.
Two people whom I reach and discuss how to do things. These are the people who are behind all this. If I'm organizing anything, the first person after the Niranjan sir is Raju sir, and then I request madam. So jealous is just a word, but they are the force behind this. Directly or indirectly. And as well as the judge, on behalf of the department and on behalf of our students, I request Ravi to have our open up objection. Motivate our students with his input.
ನಾವು ಇಷ್ಟಪಡ್ತೀವೋ ಬಿಡ್ತೀವೋ ಎವ್ರಿ ಮೂಮೆಂಟ್ ನಾವೆಲ್ಲರೂ ಬೆಳಿಗ್ಗೆ ಒಂದು ಲೊಕೇಶನ್ ಮಾಡಿದ್ವಿ ಮತ್ತೆ
ತುಂಬಾ ಚೆನ್ನಾಗಿತ್ತು. ಇದು ಸೈನ್ಸ್ ಬ್ಲಾಕ್ ಕೇಳ್ತಾ ಇದ್ರು.
ತುಂಬ ಚೆನ್ನಾಗಿ
ಯಾವ ಹಾಡು ಅಂತ ಇಡೀ ಸೈನ್ಸ್ ಬ್ಲಾಕ್ ರಿಜಿಸ್ಟರ್ ಆಗೋದಿಲ್ಲ.
ನ್ಯಾಷನಲ್ ಅವಾರ್ಡ್
ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರ. ಏನಂದ್ರೆ ನಾವಿವತ್ತು ಮೈಕ್ರೋಝೋನ್ ಅಂತ ಮಾಡಿ ಮೈಕ್ರೋಝೋನ್ 3.0 ಅಂತ ಮಾಡಿ ಸರ್ ನಮ್ಮನ್ನೆಲ್ಲ ಸೇರಿಸಿರೋದೇ ಒಂದು ದೊಡ್ಡ ವಿಷಯ, ನಮ್ಮ ಬ್ಯಾಚ್ ಎಸ್ಪೆಷಲಿ.
The first prize goes to Chaitra Ayam. prize goes to Nirmala Bai. Congratulations all the